Hello everyone, welcome to today's video lecture. Today we'll explore two key techniques used to diagnose viruses: hemagglutination assays and rapid diagnostic tests. By the end of this lecture, you will gain a clear understanding of the principles behind hemagglutination assays as to how they detect viruses, the advantages and limitations of hemagglutination assays. You will also learn the concept of rapid diagnostic tests, their diverse applications in rapid diagnosis of viruses. Hemagglutination is used for the diagnosis of some enveloped viruses such as influenza viruses. This method relies on the specific feature of some enveloped viruses that can adsorb to red blood cells. Hemagglutinin, an enveloped glycoprotein of some of the enveloped viruses, such as the influenza viruses, imparts this property. Hemagglutinin proteins on the surface of the influenza virus can bind to red blood cells and glue them together, forming a lattice-like structure. This is known as hemagglutination. This picture provides an example of hemagglutination. This shows what hemagglutination looks like in the well of a microtiter plate. Hemagglutination inhibition assay. The hemagglutination inhibition test works by measuring how well antibodies bind to the hemagglutinin proteins and prevent them from gluing the red blood cells together. That is hemagglutination inhibition. When antibodies are pre-mixed with the influenza virus followed by RBCs, the antibodies will bind to the influenza virus antigens that they recognize, covering the influenza virus so that its hemagglutinin surface proteins can no longer bind to RBCs. The reaction between the antibody and the virus inhibits, that is, prevents hemagglutination from occurring, which results in hemagglutination inhibition. This is why this assay is called a hemagglutination inhibition HI test. This figure shows what hemagglutination inhibition looks like in the well of a microtiter plate. Hemagglutination inhibition titer assay. For this, obtain a preparation of the virus, example influenza virus, with a known hemagglutinin titer, or determine its hemagglutinin titer. Prepare two-fold dilutions of the patient test serum to be tested. For example, from 1:10 to 1:20,480. Add a fixed amount of virus to every well of a 96 well plate equivalent to 4 hemagglutinin units. However, this varies according to the virus being tested. Do not add hemagglutinin to the serum control wells. Allow the plate to stand at room temperature for 60 minutes. This time varies according to specific requirements. Add red blood cells and incubate at 4 degrees centigrade for 30 minutes. Read the wells. This virus has a hemagglutination inhibition titer of 1280, which means that the greatest dilution of antibody that still blocked hemagglutination from occurring was at 1280 dilution. At these dilutions, the antibodies were still capable of recognizing and binding to the antigens on the virus. The highest dilution of the serum antibody that prevents hemagglutination is called the hemagglutination inhibition titer of the serum. Antigenic characterization assay. In this example, the hemagglutination inhibition test is used to compare currently circulating viruses rows B and C with the vaccine virus row A. This allows scientists to quickly determine if a virus used in the seasonal flu vaccine is antigenically similar to circulating influenza viruses and therefore capable of producing an immune response against them. Public health experts consider influenza viruses to be antigenically similar or like each other if their hemagglutination inhibition titers differ by two-fold dilutions or less. This is equivalent to a 2-well, that is four-fold dilution or less difference. In this example, the circulating virus 1 is compared with the vaccine virus. Circulating virus 1 differs by one dilution, that is a two-fold difference, and therefore is like the previous season's vaccine virus. However, circulating virus 2 differs by five dilutions, differs by five dilutions, a 32-fold difference, and therefore is not like previous season's vaccine virus. Circulating viruses that are antigenically dissimilar, that is, not like to vaccine virus, are considered low reactors. Advantages of hemagglutination assay. It is simple to perform, not as expensive as many other tests. The types of equipment used are generally easily available, and one can get fast interpretation of results. Limitations of hemagglutination assay. Faults in incubation time and concentration of RBC may lead to wrong results. Different factors considered in the reaction must be specific. Their non-specificity may result in an incorrect result. Determination of quantitative values and result interpretation require qualified individuals. Result interpretation is made manually without any digital data, so the different observers might have errors or fluctuations in analysis. Rapid diagnostic test definition. The World Health Organization defines a rapid test as an assay that is designed for use where a preliminary screening test result is required and is especially useful in resource-limited countries. Characteristics of rapid diagnostic tests. These tests are characterized as being high quality, easy to use tests for use in a resource poor setting, quick and easy to perform and requiring little or no additional equipment, designed for use with a single or limited number of samples, making them more economical than ELISA in low throughput laboratories, possible to store at room temperature for an extended period, able to give same day results, thus providing timely treatment interventions. The acronym ASSURED, which stands for affordable, sensitive, specific, user-friendly, rapid, and robust, was outlined by the World Health Organization to summarize the set of criteria that the ideal rapid diagnostic test should have. Here is a schematic workflow comparing the laboratory-based testing and rapid diagnostic testing. Laboratory-based tests and rapid diagnostic tests are both valuable tools in diagnosing disease, but they differ significantly in their workflow, turnaround time, and accuracy. Laboratory-based tests, samples such as blood, swab, tissues undergo a multi-step process involving transport, processing, analysis with special equipment such as microscopes, analyzers, interpretation by trained personnel, and result reporting. This can take hours to days. Rapid diagnostic tests. Samples are analyzed directly at the point of care, which can be doctor's office or clinic, using simple devices like test strips or cartridges. Results are typically available within minutes to half an hour. A lateral flow assay works similarly to a chromatographic assay where the components of liquid sample are transported by capillary action inside a paper-based membrane. A lateral flow assay strip has four main areas: the sample pad, where the sample is dropped; the conjugate pad, an area where biorecognition elements antibodies are combined with labeled tags; test and control lines, where color change indicates a positive or negative result; and the absorption pad, where excess of sample and reagents are collected. Two main types of immunoassays can be prepared using lateral flow assays: the sandwich assay and the competitive assay. The sandwich assay is used to detect larger analytes with multiple antigenic sites. In this format, the labeled antibodies are immobilized on the conjugation pad. The label can be a metallic nanoparticle or a quantum dot. Once a sample is dropped, it is transported by capillary action. When the liquid sample passes through the conjugation pad, it rehydrates the labeled antibodies attached to the antigen epitopes. The labeled antibodies attached to the antigen continue traveling along the LFA membrane and reaches the detection and control lines. If the antigen of interest is present, a colored line will appear at the detection lines. The color intensity at the test line is directly proportional to the analyte amount present in the sample. Regardless of the quantity of analyte in the sample, an anti-species antibody at the control line will bind to the nanoparticle, yielding a solid control line signal, which demonstrates that the assay is functioning correctly. When testing lower molecular analytes with a single antigenic site, the competitive lateral flow assay is used. In this format, the test line typically contains the analyte antigen and the conjugate pad contains the detection antibody labeled conjugate. If the target antigen is present, the antigen will bind to the conjugate and will prevent it from binding to the test line's antigen. If the antigen is not present, the conjugates will bind to the analyte at the test line, yielding a signal. The signal intensity is inversely proportional to the amount of antigen present in the competitive format sample. As in sandwich assay, the control line will bind the nanoparticle conjugate with or without the antigen, providing confidence that the assay is working correctly. In this case, only two types of antibodies are present in the lateral flow assay: a primary antibody coupled with labels and a highly specific to the sample antigen, and a second species specific anti-immunoglobulin antibody located at the control line. Rapid diagnostic tests are point of need devices ideal for testing outside a dedicated laboratory space by personnel with minimal training. They are low cost, require no or minimum energy, and can provide results within 5 to 30 minutes. Rapid diagnostic tests are vital tools for rapidly detecting disease outbreaks, evaluating vaccine effectiveness, performing surveys of pathogen prevalence, and allowing remote diagnosis in organized workforces entering endemic areas. Now, let us look into some specific applications of rapid diagnostic tests in virology. Rapid tests can diagnose flu within minutes, allowing for early treatment and prevention of further spread. This is particularly crucial for high-risk individuals like children, elderly, and pregnant women. During the pandemic, rapid tests played a vital role in identifying infected individuals and facilitating isolation measures. This helped slow transmission and manage public health resources effectively. Rapid tests can also provide quick results for HIV infection diagnosis, encouraging earlier intervention and access to treatment. This can significantly improve an individual's health outcome and reduce the risk of transmitting the virus unknowingly. Some kits can give accurate results of up to 99.8%. Now, let us look into some limitations of rapid diagnostic tests. False False positives. While uncommon, a rapid test may indicate a viral reaction when you are actually healthy. This can lead to unnecessary anxiety, isolation, and potential further testing. Appearance of false negatives. This is a more common concern. A rapid test might give a negative result even if you have a viral infection. This can provide a false sense of security and lead to unknowingly spreading the virus to others. Factors like the stage of infection and user error can influence false negatives. Which of the following is the principle behind hemagglutination tests? A. Virus agglutinate clump red blood cells. B. Antibodies agglutinate the red blood cells. C. Red blood cells lyse burst in the presence of viruses. Or D. Viruses replicate in red blood cells. Answer is A. Viruses agglutinate clump red blood cells. What type of virus is commonly diagnosed using a hemagglutination assay? A. HIV. B. Influenza virus. C. Hepatitis B virus. Or D. Herpes simplex virus. Answer is B. Influenza virus. A hemagglutination inhibition test detects A. The presence of viral particles in the sample. B. The presence of antibodies against a virus in a sample. C. The infectivity of a viral sample. Or D. The type of red blood cells a virus can infect. Answer is B. The presence of antibodies against a virus in a sample. Which of the following is not typically used in a rapid diagnostic test for a virus? A. Viral culture. B. Antigen detection. C. Antibody detection. Or D. Lateral flow immunoassay. Answer is A. Viral culture. Thank you for your kind attention.